Everybody has all their food? Very good. Welcome to the pre-introduction. Welcome again. We have new people here, and it looks like you guys have already found the food, which is excellent. We will also have food at 3:10 and 4 o'clock tomorrow and Friday, so if you want to come and support fellow Florentians, please do. And then also this is stored on YouTube, and is also live cast thanks to Alex, so you can go back and watch Cindy over and over and over again, if you want to. So, right, Mom, Dad. Well, Cindy came. She's my advisee and hails from San Francisco, California. It's been a wonderful 4 years. And she's doing what most Florentians are doing, which is gap years, to just figure out what to do with life and things like that. I don't know, it's kind of a millennial trendy thing. I'm not exactly sure. But it's been a pleasure. And so we will now turn it loose with the fourth beer talk in our 17 senior seminar. We the majority are beer talk. Well, a quarter of them are beer talk. Yeah, so excellent. Take it away, Cindy.
All right. Hi, my name is Guan, and today I'll be presenting my project entitled Elucidating the Roles of Zinc and Copper in Fermentation. Alright, so humans have had a long history of consuming alcohol, as you might very well know. Archaeological evidence dates the earliest firm evidence of intentionally fermented beverages to around 10,000 BC, so Stone Age. But more recently is the oldest archaeological evidence dating the creations of wines, and that would be around 6,000 BC in the area of Georgia. And even more relatively recently than that is the oldest archaeological evidence of beer, which dates to 5,000 BC, around there, and that is found in the area of Iran. Humans have also used alcohol in a variety of other purposes, such as in medicine or in religious ceremonies, but my focus of this talk today is going to be about alcohol, the beverage. Alright, so when we think of alcohol beverages, modern day alcohol beverages, we usually think of beer, wine, sake, Japanese rice wine, and spirits, which are created via distillation. But in terms of wine, beer, and sake, they're all produced via fermentation processes. We have this starting material, grapes for wine, barley for beer, and rice for sake. And the starches in those starting materials are converted to alcohol via fermentation, and more importantly, with the help of our little friend, yeast. Yeast is a very interesting organism. We use that in a lot of biological studies because it's a model organism. It's a single-celled eukaryote, so it's similar to humans, but more simple and smaller. It's very well characterized, so we know a lot about its genome, its characteristics, its proteins and such. And it has a very short lifespan, so it can be grown easily and quickly in the lab, and we can look at multiple generations of yeast very quickly. So it has a very diverse range of applications in such fields such as functional genetics, where we look at gene and protein interactions. We can also use yeast to study things like the cell cycle and tangentially aging processes. And also we can look at metabolic studies using yeast. And so, where is yeast exactly in alcohol creation processes? Well, let's take a look at the beer brewing cycle. It's a little bit small, you don't mind the words, but we start off with grains for beer, and then the starch is extracted from the beer in the milling step. Then in the mashing step, the extracted starch is put in with warm water so that the enzymes in the grain are going to convert the starch to sugars. Then in the boiling step, guess what happens? The liquid is boiled. So this does two things. Well, first, this sterilizes the beer. So when you drink it, you don't, get sick because of various microorganisms that might be in there. Also, in this step, you add hops, and that's going to impart a lot of the flavors that you think of when you're drinking beer. Finally, the next step is where fermentation occurs. So this is where the yeast is pitched, thrown in, And at controlled temperatures, the yeast is allowed to eat up the sugars and convert that into alcohol. Finally, all those particulate matters in the beer are filtered out, and it's packaged and sent to your mouths. So when we think about beer types, we usually think of three different kinds. We have ale, lager, and Belgian beer. And these can be distinguished by the type of yeast that are used in the beer. So for ale, that's going to be representing the majority of the beer types that are sold today. And the yeasts that are used in ale are going to be top fermenting yeast. So that means that in the fermentation step in the beer brewing process, they produce this piddly, squishy foam that sits on the top of the liquid. With the lager beer in the fermentation process, they don't do that, so they're called bottom fermenting yeast. And the Belgian beer use different kinds of yeast, but what distinguishes the Belgian beer from the ale and lager beer is the beer brewing process itself, which I won't go into the details of. You can imagine fun stuff. So a question we'd like to ask when we're thinking about beer is, how can we make it better? Well, brewers have several beliefs, well, many beliefs actually, on how they can improve fermentation. And here are just several techniques that they use to do so. One thing that some brewers believe is that adding zinc supplements improve yeast vitality. Now, what do I mean by vitality? It means that, well, here's an analogy. Think about you as a worker. You're gonna get tired after a long day of work, right? Well, yeast get tired too when they ferment sugar to alcohol after a long day. So zinc supposedly will supplement the yeast so that they can convert sugar to alcohol faster after a long day of work, okay? Now, Germans like to do something special where they do the boiling process in copper kettles. Copper itself has there's a physical, chemical reason why copper is great for this boiling step, and that's because copper is a good conductor. So it can evenly and quickly redistribute the heat during the boiling step into the liquid, and that makes for a good quality beer. But is there a more biochemical basis for why these things might be good for improving fermentation? Well, in order to answer this question, I would have to talk about fermentation in terms of a biochemical sense. So, this is fermentation, the metabolic process. We start with glucose or sugar, and then we go through the step called glycolysis, which converts the glucose into pyruvate, as well as ATP, an energy-rich molecule, and NADH, which is an electron carrier. For fermentation, there needs to be an absence of oxygen in the cellular environment. And so when there's an absence of oxygen, the pyruvate gets converted to carbon dioxide, and that contributes to the carbonation of the beer. So that's why you get that nice, fizzy, refreshing feeling. And is also converted into acetaldehyde. And then with the help of NADH, the electron carrier, the acetaldehyde is converted to ethanol or alcohol. Another thing that helps in this final step of converting acetaldehyde to alcohol is something called alcohol dehydrogenase. And you might be wondering, what is alcohol dehydrogenase? Well, I'm here to tell you that it's an enzyme. What is an enzyme? It is a protein that helps a biological reaction take place. And sometimes enzymes will help too with something called cofactors. And cofactors are non-proteins, but they aid the enzyme in its action, basically. So as an example, NADH is a cofactor in that last step of converting acetaldehyde to ethanol. And also, interestingly, zinc and copper are also cofactors. In this case, zinc is a cofactor for our alcohol dehydrogenase. So if you look at this little diagram, it's here helping convert the acetaldehyde to ethanol here. And then copper is going to be a cofactor for another enzyme called cytochrome C oxidase. And this isn't involved in fermentation, but a different metabolic process. It's involved in a metabolic process called respiration. So if you recall, I mentioned that glucose is converted to the pyruvate in glycolysis, and then in the absence of oxygen is converted to an energy-rich molecule in fermentation. But when there is oxygen present, the pyruvate is shuffled instead into the mitochondria, where it goes through something called the Krebs cycle, pops out some carbon dioxide, and then NADH again. And then that is shuffled through something called the electron transport system or transport chain to produce ATP and water. And respiration produces a lot more ATP than fermentation does. So here's just a little close-up of the electron transport chain and how that works. It looks a little bit complicated, but I don't want you to think or look too closely at what's going on otherwise. You could spend a while. But you have your NADH, an electron-rich molecule, and it's broken down into NAD plus and protons and electrons. And those electrons help pump the proton across the mitochondrial membrane. And then the electrons shuffle along through these various protein channels, 1, 2, 3, and 4, and more electron-carrying molecules are broken down, and those protons are then going to be pumped across these different channels. And then finally, at the last step, ATP is going to be produced. So what I want you to take away from this is that cytochrome C oxidase, the enzyme, is working here at this 4th protein channel during this electron transport chain. And that's where the copper, as a cofactor for cytochrome C oxidase, is going to work. So, if you note, at all these various junctures of these protein channels, protons are being pumped across. So you're getting a gradient of protons from one side of the membrane to the other. And so this difference in charge due to that gradient is going to be called mitochondrial membrane potential, or MMP. So keep that in mind. I'll talk about this more a little bit later. So from that information, I've hypothesized that zinc and copper increase MMP during fermentation and respiration. Okay, but keep in mind that the shuffling of protons across the mitochondrial membrane is a very dynamic process. So how are we going to measure this dynamic process? Well, I use something called Rhodamine 123, which looks a little something like this. It's a small positively charged molecule that can cross membranes like the mitochondrial membrane easily. And so it's very useful as a proton sensor. Another thing is that it's a fluorescent dye. So it is of interest to us, and I'll tell you why. Here I'm going to explain a little bit about what fluorescence is, because you might not know what that is. So this is going to be an energy level diagram where lower energy is down here, higher energy is down here. And you have a molecule that's at ground state, so it has a certain amount of basal energy, resting energy. Then you have some energy come in the form of light that excites this molecule. So the molecule absorbs this light and gains energy, and it goes into something called an excited state. When it's excited, it does a little bit of relaxation, so some of that energy goes towards the bond vibrations of the molecule. And then it goes back down from the excited state to the ground state. And in doing so, releases proton again. But this proton is going to be a different energy level than the protons that were used to excite it in the first place. And because of this difference in energy level, you can distinguish the light that's coming in versus the light that's coming out off of the fluorescing molecule. And so that's what that is. That light that's coming off of that molecule is what fluorescence is. So we measured fluorescence with something called a flow cytometer. What we do is we take our cell samples, so our yeast that have been tagged with our fluorescent dye, the Rhodamine 123, and we shuffle the cells past the path of this laser one by one. And when the laser hits the cell, it'll fluoresce and emit photons that are then redirected through these mirrors and filters through these detectors. And then those are connected to the computer, so it sends out the data that can be analyzed to the computer. And these different detectors correspond to a different range of frequencies that we can detect, or that come off from that laser, or from that excited molecule. So in our instance, rhodamine 123, we know will emit photons at a wavelength of 550 nanometers. So we're using this detector, FL1, to detect that wavelength. All right, so when we look at the flow cytometer data, we may get something that looks like this. This is a scatter plot. And on the x-axis, it's going to be the logarithmic, scale fluorescence intensity for that the FL1 detector detects. And then on the y-axis is also the logarithmic scale of a different detector, FL3, for detecting fluorescence intensity. And so you note that all these little dots are different cells. Now, when we put a sample through the flow cytometer, usually it will analyze around 10,000 cells. And so that's a lot of information from a sample, and as represented by this graph. It's really hard to condense that information into something that you can easily analyze. So how do we do that? Well, here we'll have a little histogram where, again, on the x-axis, it's going to be the logarithmic scale fluorescence intensity detected by the FL1 detector. And then the y-axis is going to be the cell counts that are fluorescing at that certain intensity, right? And you note a pretty nice, cool Gaussian distribution almost, not quite. So you can represent this. Distribution using this geometric mean, which is a measure of the central value of this distribution. All right. So going back, so how did I treat my yeast, or what did I do with that? I used 4 different kinds of yeasts. I had lager yeast, ale yeast, brew yeast, which is an all-purpose brewing yeast for beer, and then champagne yeast, which is a wine yeast. And then I stuck them in a nutrient-rich broth that they were allowed to grow, replicate, reproduce in. YPD, which contains glucose, which is a fermentable carbon source, or YPG, which is going to contain glycerol instead of glucose, but is otherwise identical to YPD. And that's a non-fermentable carbon source, so that presumably forces the yeast to respire rather than ferment. And then that is going to be left as is, or the broth is going to be supplemented with either zinc or copper at a concentration of 0.01 grams per liter. And then after that, we washed the yeast and stained it with the rhodamine 123 fluorescent dye, and then put the samples through the flow cytometer. And so you might be asking, what's the relationship between rhodamine 123 fluorescence and the mitochondrial membrane potential? Here's a little graph to illustrate what's going on. Well, the blue represents lager yeast and the red represents the ale yeast, and you've got the YPD here, and then YPD with the addition of sodium azide over here. And then the y-axis is going to be the geometric mean fluorescence intensity. So in the nutrient broth alone, we have a fairly low fluorescence intensity for both the lager and the ale yeast. But with the addition of sodium azide, we have an increase in the fluorescence intensity. Why is that? Well, I'd have to explain what sodium azide is first. Sodium azide is a very toxic chemical. Don't play with it at home, kids. It will hinder the mitochondrial metabolic processes that are occurring in the yeast. So what you're seeing here, at least with the AO, when you see this dramatic increase in the fluorescence intensity due to the addition of the sodium azide, that's probably because I've killed a lot of the yeast cells. So they're not. And then with the lager yeast, you also see this increase in the fluorescence intensity but not so much as this dramatic increase here. So, to summarize, the rhodamine fluorescence and the mitochondrial membrane potential have an inverse relationship. Okay? Now let's look at the data. First, with the zinc supplementation, all the different colors represent the different yeast species I've used. And again, on the bottom are the various nutrient broths and supplementations. And on the y-axis, it's going to be the geometric mean fluorescence intensity. And so, if you look here, overall with the YPD, so the group that is supposed to ferment versus the YPG, the group that is supposed to respire, you'll note overall like a decrease in fluorescence intensity for the YPG group versus the YPD group. Also, with the lager yeast, you'll note that the fluorescence intensity is decreasing with the addition of zinc for both the fermentable condition and the respiratory conditions here. With ale and brew, you'll notice that for YPG, it increases in fluorescence intensity and decreases in fluorescence intensity for the YPG. And then for champagne, there's an increase of fluorescence intensity with the addition of zinc in the YPD, but then a decrease with the addition of zinc for the YPG. And then for copper, you'll note a similar trend with the lager in that across the board, with the addition of copper, you see a decrease in fluorescence intensity. And again, with ale and brew, it alternates between decreasing in intensity for the YPD and then increasing in intensity for the YPG. And then champagne, you see an increase in fluorescence intensity for both broth types. So to summarize, because that's a lot of information, we see an increased mitochondrial membrane potential for YPG versus the YPD broths. And as a reminder, the YPG broth is the one that induces respiration, whereas the YPD is a fermentation broth. So I presume that this is because during the respiration process, you're producing a lot more ATP than you are for the fermentation process. And because of that, there's a lot more protons being carried off, pumped across the mitochondrial membrane. And so that would explain why there is a greater mitochondrial membrane potential and hence lowered fluorescence intensity for the YPG versus the YPD. With the lager yeast, you see an improved mitochondrial membrane potential with zinc and copper supplementation for both the fermentation conditions and the respiration conditions. For the ale and brew, you see an improved mitochondrial membrane potential with the zinc and copper supplementation in the fermentation conditions, but a decreased mitochondrial membrane potential in the respiration conditions. And then for champagne yeast, you're seeing improved mitochondrial membrane potential with the supplementation of zinc in the respiration conditions, but decreased mitochondrial membrane potential with the rest. And so this might be due to the different tolerances that yeast might have for different concentrations of metals. So in the future work, I'd like to see different yeast types being used, as well as different concentrations of zinc and copper. And the implications of this work is that supplementation of yeasts can be used to optimize the fermentation process. And this is especially important for industrial brewing where time is money. And also, I didn't mention this before, but yeast also can play a very important contribution to the flavoring of the beer. So supplementation with metals could alter the metabolism enough to alter the flavor profile of that beer. So it would be fun to look into that in the future. And finally, I'd like to acknowledge Professor Paul, Professor Donahue. You guys contribute a lot to my understanding of this topic. Daniel Martin and Wayne from the stock rooms, you guys are awesome. You guys help a lot with procuring stuff. My Count 680 cohorts who were just there to really beat into me that I need to work on my presentation skills. Thank you. And Julie Jadowski and Jason Park, you guys, listen to me. Celestial in my heart. Thanks, guys. Questions? Oh, questions? You there.
So go back to your conclusion or summary slide. Whatever.
Yes, ma'am.
So why is it that you're seeing decreased mitochondrial membrane potential with the copper condition for the YPG, which is supposed to be respiring, and copper, from my understanding, is enhancing the productivity of cytochrome C?
I like that question. That's an excellent question. I'll answer that question. So, as I said, the concentration that I use for copper, I only use one concentration in this instance. And so it might be that for these yeasts, their tolerance for this concentration might be a lot less than for different kinds of yeast, like the lager, which saw an increase. So it could very well be that the concentration is too high, and that might actually prove to have a cytotoxic effect on these yeasts in particular. Answer your question?
Yes.
Can I just ask kind of an extension of that
Of course.
question? So what range, if you were going to test more concentration, what range would you like to go higher and lower, or just lower? Is this on the upper limit of what you would like to use? And then also, how did you actually introduce it? Is it, in what form did it come in? Like, is it a salt? Is it?
I introduced it in a salt, or I should probably answer this in chronological order. Okay.
However.
Well, because there is some cytotoxic effects, or what I presume to be, what I think is cytotoxic effects with some but not others, I think this would probably be more on the upper range of concentration I've used, but I would still go further up and lower as well to look at the limits of supplementation. Also, I introduced them in the form of salt, so I put them into the nutrient broth before introducing the yeast.
What's the other end?
Oh,
It's a salt, so you have
Okay.
copper, what?
chloride.
Thanks.
I tried to find sulfate, but I couldn't find stuff, so I just went with chloride.
Other questions?
I
Oh.
think I just lost my train of thought.
Oh, the train left? Oh, no, she's got it.
I have more.
Go ahead,
Oh,
Deanna.
go ahead.
Can you go back to your actual bar graphs? Maybe the copper one. So some of these bars seem not very different. Do you have any way to say if that's actually a significant difference or not?
Not with the software I was using, no. There's a statistics test that I could use to look at the distribution to see if that was significantly different. But in this case of looking at geometric mean, I can't do that. But even though they don't look that different, the numbers are different. It's
And
just a matter of the axes that I use. I think that's a problem.
if you were to, is this just like one, I know it's 10,000 cells, but it's going to be a mean of the distribution.
Yeah.
And so it is technically just one number. If you run it again and again and again, you get the same geometric mean. Do you have any idea of the sense of variance, even if it's the same sample?
No, I don't have an answer to that. But when I run it again, because I did this several times, but I can't like show different sets of data from different sample runs because of that variance in fluorescence is going to fluctuate enough that it just wouldn't make sense to compare them like that.
So you need to normalize
So I have to look at
somehow.
one entire experiment, one entire run of these things. So I have to glean any significant meaning from that, or not significant in the statistical sense, but yes.
So I think I remember my question. So you were looking at comparing respiration conditions with the glycerol and the fermentation conditions with the glucose, but how are you controlling that? Like, how are you controlling those conditions? Like, they had to be anaerobic versus aerobic conditions, so you just didn't say it, that's why I was curious.
Oh, okay, so aerobic, cap off.
Okay.
Aerobic, or anaerobic, cap on.
Okay. I mean, I just wanted to know the details, that's all.
Okay.
thank the speaker one more time. Right.
Sam, do you think you could normalize it by like looking at something to do, sorry. So if I understand it right, you're saying